Okay, so now we're going to talk about muscle tissue and muscle physiology, and this is chapter 10. All right, so the first part of chapter 10, introduction into the types of muscle tissue. This is repeat information. Remember from chapter 4 when we discussed tissues and the histology of tissues, we spoke in great detail about the 3 types of muscle, skeletal, cardiac, and smooth. So this should all be a refresher, and so this is good because it's repetitive and helps you prepare for that comprehensive final at the end of the semester. All right, so all 3 types of muscle have the common characteristic that they are able to generate force or muscle tension. So that is a function that all 3 types of muscle tissue share. And then other functions of muscle tissue, one of the ones we think about first, I think probably the first couple of functions that pop into your head when you think of muscle is generation of tension, like generating force, creating movement. Those are probably pretty self-explanatory, and you think about those first. Some other functions are maintaining posture in the body, stabilizing joints. So if you remember back to chapter 8 when I was talking about the shoulder joint, so the glenohumeral joint, the glenoid cavity of the scapula is pretty shallow, and yes, there's a labrum there that kind of helps create a deeper cavity, but overall the head of the humerus and the glenoid cavity of the scapula, it's not a very deep cavity like what you see in the ball and socket joint of the hip or the coxal joint. And there aren't as many big ligaments binding the scapula and the humerus together. Really, the shoulder joint relies a lot on the muscles that surround it to really stabilize it in place. So that's what it means by stabilizing joints. The muscles surrounding joints help stabilize them as well. Generating heat. So when muscles contract, they utilize ATP energy and they convert, and we'll see how ATP is used later on, but they convert ATP, which is chemical energy, into movement, which is kinetic energy. And anytime you have a transfer of energy from one type to another, it's never 100%. A lot of that, or some of that energy, is going to be released as heat. And so muscles are contracting all of the time. Even if you are sitting in your desk and all you're doing is sitting upright, looking at this screen and listening to my voice, if you are maintaining posture, if your body's not collapsing, right? You have muscles contracting to maintain posture and maintain a certain level of muscle tone. So muscles are contracting all day long. And that generates heat. That heat gets absorbed by your blood, distributed around the body. And so muscles help maintain, they generate heat and help maintain body temperature. And then regulate flow of materials through hollow organs. This function is going to be really geared towards smooth muscle, which you find in the walls of hollow organs. All right, so refresher, skeletal muscle. Where do we find skeletal muscle tissue? We find it in skeletal muscle organs. So make sure you know the difference between, if I say skeletal muscle tissue, it's referring to, remember, a tissue is made up of cells that are cells, primarily the same cell type, usually the same cell type, that come together that carry out an overall function, right? Now, an organ, what's the definition of an organ? It is an organ is made up of multiple tissues that come together to carry out a particular function or functions, all right? And so a skeletal muscle tissue is this right here, all right? Long cylindrical muscle fibers that are multinucleated. They run in parallel, they have striations. This is skeletal muscle tissue. A skeletal muscle organ is like the biceps brachii or the triceps brachii, the quadriceps femoris muscle group, the orbicularis oris. Like I'm naming skeletal muscles. Those are all individually a skeletal muscle organ. because you'll see later in this chapter, they're not just made up of skeletal muscle tissue, but they're also made up of connective tissue coverings. And you'll find blood vessels running through skeletal muscle organs, and you'll see nervous tissue. And so the fact that you see multiple types of tissue in a skeletal muscle organ, like the biceps brachii, that's an organ, not just a tissue. All right, so I hope you understand the difference between a tissue and an organ. All right, so skeletal muscle cells are multinucleated, and the reason for that, so you see this is a cell, this is a cell, this is a cell. The nuclei are kind of pushed to the periphery, and you'll see why in a little bit. It's because they're so full with myofibrils. They're multinucleated because early on in development, multiple muscle cells will fuse together to form one long cylindrical cell. Reason for that is because the longer these skeletal muscle cells can be, the more they can contract, the more force they can generate, right? If you have really short cells, they're not going to generate a lot of force when they contract. But if you have really long cells, you can get greater contraction, all right? So that's why you have to have a fusion of multiple cells, and that's why you get the multiple nuclei. All right, you see striations, and those striations are due to, you'll see in a little bit, the presence of actin and myosin, which are contractile proteins, and the way that they are arranged, it appears on a microscope to be alternating light and dark bands. You get that striated appearance. Okay, so it has to do with how those contractile proteins, actin and myosin, are arranged in sarcomeres. We'll see that in a little bit. And then these cells are primarily parallel. We don't really see a lot of branching here. Contractions are voluntary, so skeletal muscle can be controlled by conscious thought. If you want to have conscious control over a movement, it has to be made, or if you are able to voluntarily control a contraction, then it has to be skeletal muscle. And an example, we all know, like, okay, flex your arm at the elbow, that's your biceps brachii. You just made that happen, okay? Extend your knee joint, and you just made your quadriceps femoris group do that. Now, one that's kind of tricky when you think about it, the diaphragm is a muscle that separates the thoracic cavity from the abdominal pelvic cavity. It's involved in breathing. Breathing is involuntary for the most part. You don't have to think about yourself breathing, thank goodness. So yeah, it's under involuntary control, but right now, I want you to take a really deep breath in and a really deep breath out, long controlled exhalation. You decided to do that. You had control over that deep breath in and that deep, long exhalation out. So your diaphragm has to be made up of skeletal muscle, or else you wouldn't be able to do that. All right? Okay, so skeletal muscle, voluntary. All right. And let's see. So the skeletal muscle tissue found in skeletal muscle organs, the main function of skeletal muscle tissue is to move the body throughout the environment. And in order for that to happen, these skeletal muscle cells can contract all day long, but in order to actually move the body throughout the environment, you have to have connective tissue connecting that skeletal muscle, those skeletal muscle cells together, and then that skeletal muscle organ to the skeleton itself. All right, this should all be repeat from chapter 4. Cardiac muscle. This is also a striated form of muscle tissue, so that actin and myosin, those contractile proteins, are also arranged in sarcomere form, where you can see alternating light and dark bands. Cardiac muscle, only found in the heart. So what's the location of cardiac muscle? It's in the name. It's only found in heart. Now, the cardiomyocytes, if you ever hear the term myocyte, M-Y-O-C-Y-T-E, that's muscle cell. A cardiomyocyte, if you look at these cells, they're much shorter, so they're not nearly as long as skeletal muscle cells. They don't have to be. And you'll also notice that for the most part, they're uninucleate. There might be a couple of cardiomyocytes that have multiple nuclei, but on average, they're uninucleate, so one nucleus per cell. And you'll notice that the cardiac muscle cells aren't nearly as parallel aligned like you see in skeletal. Rather, you see branching. And that has to do with the type of contraction that happens in the heart. The heart is it's not spherical, but it's an organ. It fills with blood, and it has to contract synchronously and pump blood up and out through the heart. And so it has more of a whirling pattern to help squeeze those ventricles to help pump blood out. So the branching pattern has to do with the type of movement that it has to carry out, that pumping of the blood. Another identifying feature of cardiac muscle cells are these intercalated discs. So these are more prominent striations, and they are actually gap junctions. So this should be repeat from chapter 4 as well. Specialized gap junctions that allows for a much, much faster conduction of an electrical conduction of the heart. So it allows for a very synchronous contraction. The faster we can have an electrical impulse through the heart tissue, then the more synchronous those contractions can be. And that makes sense for where it's located. So in your heart, you have upper chambers, the atria, lower chambers, the ventricles. Your atria have to contract together. Your ventricles have to contract together. And if they don't, then you don't want patches of your heart contracting at all different times. That's actually a condition called fibrillation, but that is not efficient for pumping blood. So you want a very synchronous contraction, and we get that because of these specialized gap junctions called intercalated discs, all right? So just like from chapter 4, you should be able to tell me, the structure of these muscle tissues, where do we find them, and why? Again, that's repeat from chapter 4. Last type of muscle tissue is smooth muscle tissue. We do not see striations here. Now, smooth muscle cells will still have actin and myosin, which are those contractile proteins, but they are not arranged in sarcomeres, so you do not see striations. They're still there, though. They just aren't arranged in sarcomeres. All right? Now, smooth muscle cells are shorter. They're kind of spindle-shaped, meaning they taper off at the edges and have more of a, oval expanded center region. More oval, centrally located nucleus. They are uninucleate as well, meaning one nucleus per cell. We find smooth muscle, and the reason for this shape is when we talk about smooth muscle, we find it in the walls of hollow organs, like the stomach and the small intestine, the large intestine, the esophagus. We even see a little bit of smooth muscle in the trachea and some of the larger airways and in blood vessels. And the type of contraction that smooth muscle carries out is a special type. If you've ever heard of the term of peristalsis, but it's a wave-like contraction that propels some sort of substance in a hollow tube forward, okay? And so that's what they do. So their shape, this shorter shape, is fine for that. All right. So again, you find smooth muscle in the walls of hollow organs, and then also eye. The skin, that would be those erector pili muscles, right? Remember that smooth muscle, that's made up of smooth muscle. And then some larger glandular ducts will have smooth muscle, and that's so that when the smooth muscle under involuntary control, when it contracts, it can secrete the glandular product. So involuntary contractions, I didn't mention that for cardiac muscle, but I hope you all know that your heart pumps without you having to think about it. So cardiac muscle is also under involuntary control. So that's the autonomic nervous system. You don't have to worry about it. Same thing with smooth muscle. You don't have to think about the blood flowing through your arteries and veins. You don't have to think about the food moving through the small intestines or the large intestines, the uterus, Those are all areas where you will find smooth muscle, okay? And you also see gaps, so many link to one another. So these smooth muscle cells will also be linked by gap junctions, allowing for a more synchronous contraction. But those gap junctions just don't look like these intercalated discs. But the same principle applies. The gap junctions allow for a faster conduction of the electrical impulse that causes contraction, allowing for a more synchronized contraction. So properties of muscle cells. I never ask listing questions like this. This would be maybe a multiple choice question, seeing if you understand what these properties mean, being able to properly identify the contractility with what that means. Or if I give you this definition, you be able to tell me which property of muscle cells it is. But I have never asked, list all 5 properties of muscle cells. Okay? There are more important things that I could possibly ask than a listing question like this. Contractility. First and foremost, contractility, the ability to contract. So that is really the movement is that contractility. So the ability to contract, proteins in the cell draw closer together, and I will show you what that looks like on sarcomeres in just a little bit. Excitability. This property just means that muscle cells have the ability to respond to a stimulus, like a chemical stimulus. We will see this so motor neurons will release a neurotransmitter. That neurotransmitter is a chemical that causes muscle contraction. Mechanical stretch, this is like in your intestines, and when your intestines stretch because of a food bolus, that stretch causes the smooth muscle to contract to propel that food forward. So that's what mechanical stretch means. The stretch of smooth muscle can actually cause it to contract. Or local electrical signals as well. And you probably know that if you've ever had electrodes put on you, or if you've seen a video of someone with electrodes put on you, and then all of a sudden you have muscle spasms, or your muscle, wherever the electrode is on, as soon as you conduct an electrical impulse through that electrode, all of a sudden that muscle is contracting and you're not controlling it. That's what that means, okay? All right, so muscle cells they have the ability to contract. They are excitable. Conductivity, this means they have the ability to conduct an electrical potential or an electrical change across the entire plasma membrane. And we'll see this later when we talk about the muscle physiology of muscle contraction. So the entire length of the plasma membrane has the ability to conduct an electrical change. So they are conductive. Extensibility, meaning they have the ability to be stretched without rupturing. So up to 3 times the resting length. All right, so muscle cells can be stretched quite a bit without actually rupturing. That's unique. Not a lot of cells have the ability to do that. Okay, so that's extensibility. Elasticity is the ability to return to the original length after that stretching. So elasticity and extensibility will often get mixed up. Extensibility means the ability to be stretched. That's all that means, is it can be stretched up to 3 times its resting length without rupturing the cell, which is impressive. Now, if it didn't have elasticity, the extensibility part would mean it gets stretched and then it stays stretched. Elasticity, that property means that once it is stretched, it has the ability to recoil back to its original length. So these are some properties of all muscle cells. All right, myocyte. So that's muscle cell terminology. Myo meaning muscle, cyte meaning cell. And so let's talk about the features of muscle cells. So sarcoplasm, this is a particular, sarco means flesh, but really referring to, if we think about what do you mean by flesh, with, usually it's like muscle. So sarco, anytime you see sarco, it's meaning flesh, or think muscle. So sarcoplasm, this is the cytoplasm of a muscle cell. Sarcolemma, this is the plasma membrane of a muscle cell. Sarcoplasmic reticulum, or SR, this is a modified or a specialized form of the smooth endoplasmic reticulum in particular. So sarcoplasmic reticulum is a specialized version of the smooth endoplasmic reticulum, which has the function to store lots and lots of calcium. And you'll see why that's important later. You'll see, and I'll point out, so here this is a myocyte. The bulk of what you see inside of the myocyte, these cylindrical organelles, these are myofibrils. Myofibrils are made up of primarily actin and myosin, which are the contractile proteins, along with other regulatory proteins. You'll see what those are. So lots of myofibrils that just jam-pack the inside, the sarcoplasm of the cell. Here's that nucleus pushed to the periphery because the bulk of what's inside are just all of these myofibrils so that it can contract. This blue stuff, this blue tubular organelle that wraps around the myofibrils, that's the sarcoplasmic reticulum. And if you look back to your very beginning of the semester notes, and we're talking about cellular organelles, the smooth endoplasmic reticulum is that tubular network that's involved in the endomembrane system. That's what that is. It's a specialized form of the smooth endoplasmic reticulum. Stores lots and lots of calcium. All right. So myofibrils, I've already talked about that, but as cylindrical organelles, 50 to 80% of the cell volume are these cylindrical organelles called myofibrils. Again, primarily made up of actin and myosin, which are those contractile proteins. Now, other organelles that you see packed around these myofibrils are mitochondria. So you see lots of mitochondria. That's actually why muscle looks reddish or appears red. It's because it has lots of mitochondria. Why? Mitochondria are the powerhouses of the cell. They produce lots of ATP, and that ATP you'll see is needed for muscle contraction. So it makes sense that lots of mitochondria would be located around these myofibrils. Also makes sense that smooth endoplasmic reticulum, the sarcoplasmic reticulum, would be surrounding the myofibrils because calcium is also needed for muscle contraction. And the sarcoplasmic reticulum stores lots of calcium. Okay? And just remember that the myofibrils, so smooth muscle cells, myofibril arrangement is different than in cardiac and skeletal muscle cells. That's why you see striations in cardiac and skeletal, but not in smooth muscle. It's because they are arranged differently. They are irregularly arranged in smooth muscle versus arranged in sarcomeres and all along the length of cardiac and skeletal. Let's see. Want to make sure that okay. I probably got a little bit ahead of myself, so I might repeat a few things. But here, structure of a skeletal muscle fiber. Again, they're very long cylindrical cells, multinucleated because they're formed by a fusion of multiple myoblasts. Myoblast just means a very early developmental form of myocytes. I'm not going to ask what a myoblast is, but that's what that is. So skeletal muscle tissue consists of many skeletal muscle fibers. And when you hear the term, let me clarify what some terminology stuff. Myocyte, skeletal muscle cell, skeletal muscle fiber, all means the same thing. A lot of people will use them interchangeably. Skeletal muscle fiber does not mean that it's not a cell. You might think protein fibers like collagen and elastic fibers. Those are protein fibers. Those are not cells. A skeletal muscle fiber is called that just because of how long and cylindrical they look like fibers, but they're actually cells. So please don't get that confused. It's not the same as like a collagen fiber or an elastic fiber or a reticular fiber. Those are protein fibers. This is a skeletal muscle cell or a skeletal muscle fiber. All right? And I'll show you what the endomysium is in just a second. All right, so length up to 30 centimeters, thickness up to 100 micrometers. I'm not going to ask those numbers. What I want to point out is just for comparison, this should be somewhat to scale. Size comparison, this is a red blood cell that flows through your vasculature. This is a skeletal muscle cell, one cell. All right, so just to show you the size and length comparison. Makes sense, skeletal muscle fibers are found in skeletal muscle organs. The longer these cells are, the more contractile force can be generated. All right, so this is just showing here's a bone, tendon, which connects our skeletal muscle. This entire thing would be a skeletal muscle organ. So it's pulled out. If you take a cross section through skeletal muscle organs, just imagine this is the biceps brachii. The cells are arranged in bundles called fascicles. So this is an entire fascicle, which is made up of lots of skeletal muscle cells. And so here, this is an individual skeletal muscle cell. All right. So again, you'll see bulk of the inside are those myofibrils, made up of primarily actin and myosin, contractile proteins, along with some other regulatory proteins. Some other structures I want to point out, I've already talked about the sarcoplasmic reticulum. That's that specialized smooth endoplasmic reticulum which wraps around those myofibrils. It's the blue tubular organelle, stores lots of calcium. That's what's important here. The structure that is located in between, so blue, this is a sarcoplasmic reticulum. This is sarcoplasmic reticulum. These expanded areas right here are called terminal cisternae of the sarcoplasmic reticulum, and you should know that. The expanded areas right here of the blue organelle, that's called terminal cisternae of the sarcoplasmic reticulum. Now, notice they sandwich a structure in the middle, and what they are sandwiching is called a transverse or T tubule. So you can call it a transverse tubule, you can call it a T tubule. And I'm actually going to go to, let's see, I think I have I'm going backwards. I'm so sorry. Okay, there. There's my larger image. I was hoping that you could see it a little bit better. So here's our transverse or our T tubule sandwiched in between the two terminal cisternae of the sarcoplasmic reticulum. And look right here, this outer yellow structure, this is the sarcolemma. It's the plasma membrane. It is the plasma membrane of the skeletal muscle cell. And it dips down. Look, it invaginates and dips down and forms a transverse T tubule. So keep that and remember that. The transverse T tubules are invaginations of the plasma membrane, the sarcolemma. They also wrap around the myofibrils. They are sandwiched between two terminal cisternae of the sarcoplasmic reticulum, which stores calcium. So this is a structure of the plasma membrane. These terminal cisternae store calcium. And so over here, look, this is just a magnified image of that. Look, this is even better. So opening of the T tubule. Here's our sarcolemma, the plasma membrane. It invaginates in, forms this T tubule, which wraps around the myofibrils. These expanded regions of the sarcoplasmic reticulum are called the terminal cisternae. Together, this makes up a triad. All right, and you'll need to know that later for muscle physiology. All right, nucleus pushed to the periphery, mitochondria sandwiched in between the myofibrils. Now, the connective tissue covering. All right, so here's our plasma membrane, the sarcolemma, surrounding and ensheathing the skeletal muscle cell, or the myocyte, is a connective tissue investment called the endomysium. The endomysium is a loose areolar-ish type connective tissue, so it's going to be vascularized, which is good. Skeletal muscle cells are going to need a lot of oxygen and nutrients because they contract a whole lot. All right, so let me go back to this and just make sure I got all the things I wanted to say. I've already mentioned myofibril several times, sarcoplasmic reticulum several times. Key here, sarcoplasmic reticulum stores calcium. Calcium is required for muscle contraction. And again, we're focusing now on skeletal muscle cells or skeletal muscle fibers. Transverse T tubules, I already talked about that. It's a tunnel network. It's invaginations of the plasma membrane. So again, the inside of the T tubule, right, would be continuous with the extracellular fluid, all right? Terminal cisternae, talked about that. Those are the enlarged portions of the sarcoplasmic reticulum. Together, the terminal cisternae and the transverse T tubule form the triad. All right. So yes, I hit on all the key points that I wanted to talk about. So now let's focus more on these myofibrils and how they are structured. What's the anatomy of a myofibril? All right, so myofibrils, again, are made up of primarily contractile proteins, which are going to be actin and myosin, and their job is to generate tension, all right? That's why they are contractile proteins. There are also regulatory proteins involved, and it's in the name. These are going to be proteins that regulate muscle contraction. That's going to be troponin and tropomyosin. I'll mention that again later. Just hold on. And then structural proteins, which the structural proteins are primarily involved in just keeping the myofilament arrangement stable, all right? So they just kind of hold everything in place, because you can imagine that if myofibrils, which are made up of lots and lots of myofilaments, all right, so that term myofilament is referring to actin and myosin. Those are filamentous proteins. So here, this whole thing is a myofibril, and these myofibrils are composed of myofilaments called actin and myosin, all right? Just to clear that up or make sure you understand that. The myofilaments are going to be primarily actin and myosin, which are the contractile proteins. Regulatory proteins, this is going to be troponin and tropomyosin. They regulate the contraction. And then structural proteins just kind of hold everything in place and make sure they are properly aligned and stable. All right, so thick filaments, this is myosin. Thin filaments, actin. Actin and myosin are contractile. Tropomyosin and troponin are regulatory. And there are elastic filaments, and I'll show you there's one massive spring-like elastic filament called titin. It's called titin because it's one of the largest proteins as far as the number of amino acids that make up that particular protein, so that's why it's called titin. It's really, really large. Stabilize it, and this helps stabilize myofibril structure and resist excessive stretching. So that's when we think about that elastic component to muscle cells, it's because of that titin has the ability to recoil back to the original length. So let's see what these look like. All right, so here, this is a myofibril made up of myofilaments. These are the myofilaments: actin, which is purple, myosin in red. Myosin is thick. Look at the difference. So if we look, especially over here in this diagram, thin filaments are actin, thick filaments are myosin. So over here, this is myosin enlarged. Filamentous proteins with globular heads on the end, and that's going to be important when we talk about muscle contraction or the physiology behind muscle contraction. And you'll notice that these globular heads extend up and down, and they have the ability to attach to the actin above and below. So myosin thick has globular heads, long filamentous proteins with globular heads. Actin are the thin filaments, the thin myofilaments, and they are actually globular. It's made up of a globular protein. These are individual actin subunits, globular proteins, but they link together and form long filamentous strands. So they're like beads on a string, but each one of these guys is an actin subunit, and then they just link together and form this helical. So it's actually two actin strands that form like a helical structure. All right, so key takeaway, myosin are the thick filaments, the thick myofilaments, and they have these globular heads that branch above and below. Actin are the thin filaments, and on the actin is where you find the regulatory proteins. So that's a key thing to remember too. All right, what are the regulatory proteins? Troponin and tropomyosin. Troponin are these guys right here. Notice troponin is made up of 3 little subunits. Tropomyosin troponin is a globular protein made up of 3 subunits. Tropomyosin is a long filamentous protein that also forms a helical structure around the actin. Take note right here, because I'll talk about this with muscle contraction. Every actin subunit has an actin active site. This is where these globular heads will bind, all right? This actin active site will be covered up by tropomyosin. So look, if you can look in here, all those little holes, those little divots are covered by tropomyosin when the skeletal muscle is relaxed. So if contraction is not happening, then tropomyosin covers these active sites. Myosin heads cannot attach if tropomyosin covers them. Troponin is going to be attached to the tropomyosin. One of the subunits attaches to tropomyosin. Another subunit attaches to actin. And the third subunit is going to bind calcium. All right, I'm getting ahead of myself because the more you hear it, the easier it's going to be. So I'm talking about it now. I'll talk about it more later. Troponin has 3 subunits. One subunit binds tropomyosin. Another subunit binds actin. The third subunit binds calcium. And only when calcium binds troponin can it shift and move tropomyosin out of the way. So when calcium binds, troponin moves tropomyosin, exposes these active sites, and now myosin can bind. All right, so I'm saying that now because it will make it easier later hearing it. All right, so I've already, talked a lot about this. Let's see. Yes, so these globular heads, remember, will bind to the active site on actin. And then the way that they're angled, you can envision that they're going to act like a hinge. And when muscle contraction happens, these heads actually pivot inward towards this middle M line, pivot in and slide this actin in towards the center. And then same on this side too. These globular heads from myosin attach to actin, pivot and move it in. Attach, pivot, move it in, and slide this actin towards the midline. All right, so that's what it means by hinge-like neck. And then I've already talked about actin, which is the other myofilament. It's the thin myofilament. These purple subunits, those are the actin subunits. They have that actin active site. That's where those globular myosin heads bind. Tropomyosin is a long filamentous protein which covers and hides the actin active site. Troponin is a globular protein, has 3 subunits. One binds tropomyosin, one binds actin, the other binds calcium. When calcium binds, it causes troponin to move, which, since it's bound to tropomyosin, moves tropomyosin out of the way. When tropomyosin moves out of the way, it exposes the actin active site, and now myosin can bind here. All right. Troponin and tropomyosin are regulatory proteins. Actin is the contractile protein.